Good morning, everyone. Thanks for joining Viva Biotech's webinar. Today, we will explore the insights from Viva Biotech's GPCR and TPD structure biology to accelerate the drug discovery. Please allow me to introduce our two speakers today. Dr. Paul Wan, Vice President of Early Discovery and Business Development at Viva Biotech, brings 25 years of experience in drug discovery. He has a proven track record of enabling and delivering early discovery hit-to-lead projects across a broad range of therapeutics areas. Dr. Yichen Zhang, Director of Biology at Viva Biotech, leads a team focused on protein production, characterization, and crystallography. His team has delivered over 1,000 protein ligand structures in past 2 years, enabling 2 compounds to enter IND and phase 1 clinical trials. After our 2 speakers sharing, we will have Q&A section. Please feel free to type your question in the chat box. Now, I will give the time to our 2 speakers, Dr. Paul and Dr. Jerry. Please start your sharing.
Thank you very much. So while Jerry gets the slides up, I'd just like to thank everyone for joining Jerry and myself for this webinar, where we'll discuss how Viva are supporting our clients in the areas of GPCR and TPD structures. So next slide, please, going forward. Just a brief content. I'll give a background review of Viva Biotech first before moving on to target protein degradation and GPCRs. So moving forward, please. So Viva Biotech is a CRO with fully integrated drug discovery capabilities and platforms. It's really underpinned by our protein production and structure biology group, which are industrial leading. They also encompass the cryo-EM and membrane protein technologies. The protein team's been servicing the R&D community for around 15 years with high quality proteins. They also feed reagents into the capability platforms. Within the area of affinity-based screening platforms, we do a lot of our hit finding strategies with things like DNA encoded libraries and ASMS. These are also complemented by our biophysical technology platforms. So fragment-based drug discovery is undertaken by things like SPR and also our crystallography. The bioassay platforms using the in vitro screening and also cell-based assays also conduct screening and hit finding strategies. We also have a very active biologics arm. So the therapeutic antibody discovery covers things like the monoclonal antibodies, bispecifics, and Fabs, nanobodies, and SCVs. All these are actually complemented by our computer-aided and artificial intelligence drug discovery teams. So they do a lot of the structure-based virtual screening and molecular modeling for our teams. Obviously, for a CRO in small molecule discovery, we have a very capable medicinal chemistry group, particularly specializing in PROTAC chemistry and also DNA encoded library generation. Obviously, if we have a successful project, we also have full CMC CDMO capabilities. And let's not forget the DMPK team. So we have full in vitro ADME and in vivo PK studies. So moving on now to the target protein degradation. So TPDs is also known as a proximity-based modality, otherwise known as PROTAC. This is a trademarked name belonging to the company Arvinos. PROTAC stands for proteolysis targeting chimeras. The strategy harnesses the cell's own degradation pathway to remove proteins from the cell via the proteasome. So on the diagram on the left, you see around 9 o'clock of the image, the protein of interest for degradation is marked in yellow. And also you see the E3 ligase complex. The PROTAC is a small molecule, which is a bivalent molecule, which means it can bind both the protein of interest and also bind E3 ligase. At around 11 o'clock of the image, you see when this PROTAC pulls together these 2 proteins, you form the ternary complex. That's the protein of interest, the PROTAC, and the E3 ligase. And it's during this proximity where the E3 ligase ubiquitinates the protein of interest. So you can see this by the 4 red ubiquitin chains. This actually flags then for proteins like P97 to come along to bind to the protein of interest, start unfolding it, and then transferring it to the proteasome. The protein then is degraded, removed from the cell, and the PROTAC is actually recycled for further use. So in this way, you're removing disease proteins completely from circulation and the cell. Next slide, please. So Viva's target protein degradation platform has been around since 2018. And during that time, they've delivered over 90 projects, both as a fee-for-service or FT models. So about 3,500 compounds have been delivered. A lot of the proteins, the ligands, have been found by either DEL screening or ASMS. A lot of the patents have also co-authored by our scientists with clients. Since 2018, we've been building up the platform. So now the platform includes custom-built libraries. We've over now 50 E3 ligase binders, and most of these have also been conjugated to pre-linkers to allow fast follow-up once we actually get our hits. We've also generated around 120 protein of interest binders. So these are the proteins which are commonly requested in therapeutic areas. We've also managed to structurally enable around 10 of the more common E3 ligases used in therapeutic requirements. There are around 600 E3 ligases in nature. So of these 10, they include things like cereblon, VHL, which are very popular, MDM2 and TRIM24, to name a few. We've also solved a handful of novel E3 ligases. It's not really within the remit of this webinar to talk about how we work with TPDs and actually work with chemistry. However, we have produced a webinar earlier in the year. So if you look at the QR code or do a search on YouTube, Viva Biotech TPDs, you should be actually pointed to the other webinar. Next slide, please. So TPDs have really opened up a whole area of previously undruggable targets. And these are targets like transcription factors, protein-protein interactions, and also certain cancers, including carcinogens. At the moment, there are around 80 candidates in clinical phase studies. On the right-hand side, you see there's a graph. The preclinical studies seen in the blue, you see this huge increase from around 2018 of all the preclinical trials. There are also 2 or 3 very advanced phase 3 studies. Below the graph, you see the therapeutic areas where TPDs have an impact. A lot of them you see are cancers, but if you look around the outside of this image, you can see things like viral infections, epilepsy, dementia, and eczema. There are around 100 TPD biotech companies currently working, and they're thought to have raised around $10 billion in VC funding. Biotechs normally partner up with large farmers in order to actually carry out some of their research. And it's thought that if all the biotechs were to hit their milestone payments, they would trigger off a $24 billion windfall. But at the moment, there aren't any TPD drugs on the market. They expect the first one to be around 2027. These are led by the company Arvinas. They have 2 current drugs in clinical phase 3. ARV-471, which targets estrogen receptor for breast cancer, and ARV-766, which targets the androgen receptor for prostate cancer. There is a third drug in clinical phase trials, and that's the PROTAC version based on palbociclib, and this targets CDK6 breast cancer. Okay, next slide, please. So we're now going to move track to G protein coupled receptors. This is a much better researched area. G protein coupled receptors form the largest family of human membrane proteins, and they are an important class of drug targets. They regulate many of the cellular responses by initiating whole signal cascades. These are things like telling cells to proliferate, to grow, to release various cytokines. So this is very tightly regulated, and you can imagine a deregulation of this pathway can cause a wide variety of diseases, which is probably no wonder why around a third of all FDA-approved drugs targets GPCRs with an estimated $6 billion market. Historically, lots of ligands have been targeting the orthosteric site of GPCRs, but even though there are different GPCR families, some of these orthosteric sites can be quite homologous. So you get greater selectivity when targeting allosteric sites or cryptic allosteric sites. There's a third class of ligands called the bitopic ligands, and these target both the orthosteric and the allosteric sites, and you link these together. So this marries very well with the PROTACs and TPDs. So on the left-hand side is a very simple schematic of how GPCRs work. In image one, the GPCR that's inactive just sits in the membrane. When it binds a ligand in image two, such as an agonist, induces a conformational change in the GPCR, which allows it to recruit the heterotrimer complex, the G protein complex. And this is made of G proteins alpha, beta, and gamma. It's the alpha subunit which can turn over GTP to GDP, which then signals the whole cascade of messages. There are various subtypes of G alphas, and so this differentiates the type of signals it can elicit. Likewise, the G protein B can also form secondary messengers. Next slide, please. So there are many strategies to isolate GPCRs now, which Jerry will go into. And coupled with the cryo-EM revolution, this has led to a huge explosion in GPCR research and GPCR structure determinations. On the right-hand side, lot of people will be familiar with this graph. It just shows the number of cryo-EM GPCR structures that have been deposited in the EM database. As you can see from around 2019, huge acceleration in deposition of these structures. For reference, I've also put in the cryo-EM structures of E3 TPDs. Again, you can see from around 2022, there's been a huge interest in determining structures of TPDs. And the very aligned area of research, the molecular glues, I've also added those, how many cryo-EM structures of those have been put through. So Viva has been very active in the area of GPC research for many, many years, initially solving all their GPCRs by crystallography using lipidic cubic phase. And those who know about crystallization knows that this is quite a painstakingly high resource intensive activity. More recently now, routinely we're using cryo-EM to solve all our cryo-EM structures. So currently we have over 30 GPCRs structures solved. Some of these are literature precedents and some of them are very novel and new to man. Most of these are at 3 angstroms or better. So now I'm going to hand over you to Jerry, who's going to discuss the strategy, the science and technology behind the TPDs and GPCRs. So take it away, Jerry. Thank you.
Thanks to Paul's introduction to TPD and GPCR as well as Viva service platforms. So I will go into the details of these two areas from the perspective of protein preparation and the structural biology. So Viva has a well-established protein and structural platforms for targeted protein degradation to boost client research on degraders discovery. So for protein production, we have developed a full toolkit for expressing wide range of E3 ligase proteins. And we have also accumulated extensive experience in preparing stable, high-quality complexes. So for structural studies, we have a strong team of well-trained professionals who are using crystallography and EM to explore E3 ligase complex structures. So furthermore, we also have our proprietary fragment soaking system, which facilitates the discovery of new compound binding site in TPD proteins. So just as Paul mentioned, we have delivered over 140 E3 ligase complex crystal structures, including more than 10 molecular glue structures. In the meantime, we also delivered 30 cryo-EM structures. So these tables show Viva's toolbox of various E3 ligase proteins that we have produced, spanning from a cooling ring E3 ligase system to other types of E3 ligases. So our expertise in producing good samples allows us to respond to clients' requests and deliver high quality sample rapidly. So it can push projects forward much faster. So more importantly, it also demonstrates our strong capabilities of handling TPD related protein production. So we have one client with a protein production request for a E3 ligase complex. So their initial attempt to produce this complex was not very successful by following reported protocols. So they had to use over 100 liter of E. coli cells to express the protein, but only got less than 10 meg of the final product with just okay-ish quality. So you can imagine it's super laborious. However, on our hand, we actually did a lot of exploration on this target before, so we shared our experience. So with our optimized expression conditions in insect cells, we help them obtain about 20 meg of highly pure complex samples with only 3 liters of cells. So you can see that it dramatically saved workload and time. So this gallery showcased interesting crystal structures determined at Viva. So by dealing with various cases, we have developed robust methodology to maximize our successful rate to solve TPD-related complex structures. So I would like to share one case here with you. So when we were trying to identify the structure of a beta TRCP skip1 Degradation complex, it was very interesting to find that the phosphorylation status of the peptide could make crystallization behavior quite differently. So co-crystallization with a non-phosphorylated peptide did not actually yield diffractable crystals. However, the phosphorylated peptide can produce high-resolution structures even better than 2.5 angstroms. So after taking a closer look at the binding site, we found that the phosphorylated serine on the peptide contribute a lot to coordinate with the residues on the propeller region of beta TRCP. So this connection actually stabilizes a complex to form high-quality crystals. So these findings provide very useful structural information for following successful binder design. So here are the two examples of cryo-EM structures of E3 ligases in complex with target proteins. So under the microscope, we can clearly observe single particles of these complex. So it enables us to successfully deliver high resolution structures. So it should be noticed that making the complex stable is key to the discovery of full complex without losing any part. So our cryo-EM team is doing a great job to make the complex integral and stable under cryo conditions. So we are trying to make sure that the whole complex can be observed for high quality data collection. So the next session is about preparation and structural characterization of GPCRs. So I would like to share some of our experience in making GPCRs for your functional and structural studies. So as we know, GPCR is the largest cell surface receptor family. So they share conserve 7 transmemory helices, which are connected by 3 intracellular and 3 extracellular loops. Different types of substrate can trigger GPCRs to go through a dynamic conformational change to elicit downstream signal transductions. So to prepare high-quality GPCR samples for studies, we have to consider mainly 4 aspects, including construct design, expression, stabilization, and purification, and reconstitution. So to obtain high yields of GPCRs, we can do several things. So we need to design the construct very well. So we can do codon optimization and insert COSAC sequences and signal peptide sequences. For structural studies, we may consider removing flexible regions and introduce thermal stabilizing mutations. The most commonly used method to enhance GPCR expression is adding fusion tags or proteins to certain regions on GPCRs. So these fusion proteins are structurally stable. So in the early days of GPCR structural biology, T4L is innovatively fused to GPCRs for crystal structure determination. These days, we have a set of tools that can help us to stabilize GPCR and enhance their expressions. So they can be put to the N and C termini of GPCRs as well as being inserted into the intracellular loop 3. So for example, GFP when fused to GPCRs, it functional it functions not only as a fusion partner, but also as an indicator to show the expression level. For example, Braille, due to its very unique structures, is always used to insert into this part as an extension of the two helices of H5 and H6. So it potentially stabilizes the structure of a GPCR and provide more rigidity. So screening construct is necessary to identify the best clone to work with. When we are doing screening, several factors we should consider, including tags and fusion proteins, detergents, and construct lengths, as well as organism. The most straightforward way is to adopt the fluorescence-based techniques to test the construct stability and expression levels. So TSCA can help us to estimate the stability of constructs under different conditions. So fluorescence detection SAG can help us to determine the yield of GPCRs, which is the most popular way. When we fuse the GFP with our target GPCRs, we can extract the sample from the membrane and then load it to the SAC columns, and then pass through the fluorescence detectors. So the sensitivity of fluorescence enable us to capture the signal even with a little protein. And these two techniques can even be combined to screen thermostable construct with good column performance. So at Viva, we can do fluorescence-based screening through UPLC by loading GPCR samples on a plate to find the best construct and conditions. So usually two forms of GPCR can be prepared based on the purpose of your studies. So GPCR alone is the inactive form, which can be used for screening and crystallography. To stabilize the stabilize the GPCR, we can introduce a thermal stabilizing mutations and adding some fusion proteins or antibodies. With the coupled when coupled with a heterotrimeric protein complex, GPCR form the active form and can be used as for cryo-EM studies. Some factors can be added to the system to stabilize complex like nanobodies and SCFVs. So engineered G-alpha subunit like dominant inactive G-alpha and mini G-alpha can also be used. So the expression of GPCR is usually done in insect cells. Compared to mammalian cells, insect cells can produce GPCR efficiently at a lower cost. So that's why it's most widely used nowadays. By contrast, mammalian cells can provide natural PTMs and a lipid environment, which can make GPCR happy. So moreover, if we want to do the compound screening, we can establish stable cell lines using mammalian cells. However, we should know that insect cells can cause some misfolding and aggregation issues, and the virus preparation can take a long time. So the mammalian cell system may produce less protein with a higher cost. So if stable cell lines is to be made, it can take several weeks. So E. coli and yeast can also be used, but not as frequently used nowadays. After expressing GPCRs, we want to solubilize a sample from membranes. There are mainly two ways that we can use. So the first choice would be the mild detergents like DDM and LMNG. Digitoning and GDN can be used for cryo-EM studies. And the other way is to use copolymers to form nanoparticles to extract a GPCR directly. So this method is actually considered healthier for GPCRs because the natural lipid environment can largely guarantee the stability and the activity. So the purification of GPCRs rely on high selectivity tags. So his tag should at least contain 8 histidines. And we can also use tandem flag and strap tags. Some epitopes and large fusion tags can also be used to achieve better specificity. So during the preparation of GPCR complex, we can add some stabilizers to tighten the complex, like nanobodies and SCFVs. So these days, a nanobit strategy is very popular to prevent complex dissociation. GPCRs are usually tethered with the large bit, while the G beta subunit is fused with a high bit tail. So the interaction between the large bit and high bit largely improve the integrity of the complex. So GPCRs can be further reconstituted into different forms, including MSP nanodiscs, SMOPs, amphipro and seposin-based nanoparticles, and proteolipso. So at Viva, MSP nanodiscs and SMOPs are mostly made. These nanoparticles can maintain good stability and activity of GPCRs. According to some reports and our experience, GPCRs in nanodiscs can even have ligand binding activities for over 10 days at 4 degrees. Or on ice. By contrast, the same GPCRs in detergents can easily lose their activities within less 1 week. So that's why we are inclined to do nanodiscs or SMOPs for the sample, which is to be shipped out in a long journey. So we should always consider several things to achieve successful reconstitution. For nanodiscs, the size of the sample and the lipid composition, protein MSP lipid ratio should be explored. For SMOPs, if an SMA copolymer is used, we should always check the buffer pH and divalent cation concentrations. So here are the examples of construct design and expression of GPCR in different systems at Viva. So we have a GPCR platform to support the preparation of various GPCR forms. And our experienced scientists provide tailored services for studies. So there was one case, and a client came to us for a protein production request for DEL screening. Because they understood that the successful DEL screening significantly depends on the quality of GPCR samples. So after comparing Viva with several other vendors, even if he couldn't provide the same low price, they still choose to collaborate with us because they were impressed by our extremely high quality of GPCR samples. So this slide shows our capability of solving crystal structures of GPCR ligand complexes. So just as Paul said, we routinely use LCP-based techniques to produce GPCR crystals. More importantly, we have a specially developed data collection strategy for GPCR crystals. So you can see like these high-resolution structures were solved using this method. Even without too much effort to optimize crystals, we are able to identify clear ligand densities in the structures. So for the cryo-EM service for GPCRs, we are building up an effective feedback cycle between the protein preparation team and the cryo-EM team. So team can react to the project progress rapidly through seamless internal communication. Once the protein team gets the protein sample ready for cryo-EM, they will notify the EM team and book the machine. So in this way, we can always guarantee that the sample are freshly made without being delayed or damaged. So the feedback about the sample quality can also be returned to the protein preparation team based on the observation under microscopes. So this workflow makes it realistic to deliver high-resolution structures within even 3 to 4 weeks. So by taking advantage of this work model, we are able to determine cryo-EM structures of different types of GPCRs with high resolutions. Moreover, in our system, we deliver high-resolution structures of other memory proteins, including ion channels and transporters. So honestly, the advantage of incorporating GPCR production and structural determination is our signature service. So it largely keeps the fragile samples fresh and safe for EM studies. I know one case where we produce a GPCR for a client, and the client asked us to ship the accused samples to a different cryo-EM service provider. So although we notify the provider of the shipment, and they didn't respond to us right away, so we hadn't received any feedback until 1 month later. And they told us that the sample didn't behave well under microscopes. So we assumed that probably they didn't even touch it upon the arrival of the sample. So you can see that it caused big losses regarding both time and money. So one last case I would like to show is the structural identification of the ligand-bound GPCR structure by cryo-EM. So as we know, it is relatively common to solve GPCR G protein complex structures by cryo-EM. However, the determination of the inactive form is rarely reported. And in the case of for a novel GPCR, we inserted the Braille sequence into the intracellular loop 3. To make the molecule bigger, we added 2 more blocks of the anti-Braille Fab and the anti-Fab nanobody. So after screening optimal construct and expression conditions, we follow this workflow. to obtain high purity GPCR sample for EM. So from negative staining to high resolution structure determination, we finally deliver the structure of GPCR ligand compound complex at a 3.5 angstrom resolution. So we can see like here is the GPCR part with the Brill helices extruding out. And here is anti-Brill fab. And the last part is anti-fab nanobody. So it demonstrates our comprehensive capability of handling various forms of GPCR. So it potentially expands the use of cryo-EM to identify the interaction between compound and the inactive forms of GPCRs. So thank you very much for listening, and many thanks for your time. Paul and I will be happy to take your questions. Thank you.
Thank you so much for Dr. Paul and Dr. Jerry's insightful sharing. Now, let's move to the Q&A section. Please go ahead and type the questions in the chat box. The first one is, very impressive list of E3s. Just wondering, are all the E3s in the list validated to be structure biology quality?
Yeah, so I think the list that Jerry showed showed all the E3 ligases of different families that have been expressed, and a lot of these proteins would have been expressed for screening downstream. Not all of those have been in that large list have been structurally enabled. We have around 150, 160 E3 ligase structures. So chances are that if it's one of the more common E3 ligases required for therapeutic use, we will actually have those in our library. But yeah, that rather impressive list that Jerry showed was just all the proteins that we've managed to clone, express, and purify, not necessarily go all the way through to structural analysis. So they may have just been used for generating chemical hits.
Yeah, thank you for the answer. The second question is, do we have OTS samples readily available for all the E3s in the list?
So off-the-shelf sample.
Oh,
Go ahead, Jerry.
Oh, sorry. Yeah, so I can take this question because like in my team, we have produced a lot of E3 ligases related protein samples. So like for like very commonly used E3 ligases, so I can go back to, just give me a sec, I can go back to this. Oops. I cannot. Just give me a sec. So, for example, I'm not quite sure like what type of E3 ligase you are interested in, but for the commonly used system like FBW7 series, like we have a beta TRCP ready, FBW7 ready, and TRCP2 ready to go, and we also have FAM1C, FAM1B with elongin B, elongin C. And we also have 01, Zigo 11B, and KLHDC2. And in cooling 3 system, we have KLHL12 SPOPs. And for cooling 4, we have CRB and DDB1 definitely, which is the most commonly used complex. And we also have like a decaf series with DDB1 or DDA1. And we also have SOC systems. Yeah, so I just wonder like if you have some of these are of your interest. Yeah. Thank you.
What are the specific differences in preparation methods of nanodisk and the SMOPs?
All right. Okay. Thanks for your question. So I can probably handle this first. So in our experience, when we were trying to develop the MSP nanodiscs, usually we need to purify the protein in the detergent. And after getting that, we are going to disturb the sample in the detergent. And at the same time, we are going to add the MSP, the lipid, to the system. So we are going to use the BioBeads to remove the excessive detergents. And then usually we're going to use some affinity tags that are attached to the protein or the MSP to further purify the nanodiscs. So usually we can also to purify the best sample, we can also consider using a SEC column to get the final product. Yeah, and for the SMOPs, it's kind of different because just as I introduced, we can add the SMOPs directly into the membrane prep. So it's actually happened in the early stage of the purification. So without using the detergent, we can directly reconstitute our target GPCR protein into the SMOP, and it is made as a reconstituted form. So we can use the affinity tags to purify SMOPs. So without too much worry about the detergents, because we know that detergent can not be so good in some situations. Yeah. So hopefully that answered the questions.
Thank you, Jerry. We have one more question. With TPD ternary structures, how has that helped with optimization of the PROTAC being studied?
Yeah, I could probably take this one. So the PROTACs, I suppose you're looking at three aspects when you're looking at structure-based drug discovery. Obviously, you have the ligand that binds the protein of interest and the ligand that binds the E3 ligase. So on those aspects, you can probably consider using traditional structure-based drug discovery. The only difference is, while you're normally in structure-based drug discovery of small molecules, you're trying to optimize the binding, increase the affinity. With TPDs, you don't really need to worry too much about the affinity of the ligands to the compounds. I've worked on projects where we started looking for a small molecule to inhibit a compound, but we could never really improve the binding efficacy. So we decided to go through a target protein degradation pathway, and that works. So you don't really have to worry too much about the affinity of these two ligands. The critical difference with PROTAC is obviously the linker. The linker itself can be, well, it's a compound, so it also has ADME properties. So you need to consider the ADME properties of the linker, but also the rigidity. I found that simply just getting the protein of interest and the E3 ligase close to proximity is not enough to actually elicit degradation. Sometimes you don't get that ubiquitination even when these two proteins are close together. So what we found is sometimes with the linkers, the actual relative orientation of the protein of interest and the E3 ligase is critical. So it's trying to form various vectors or angles or kinks in the linker to actually change the orientation of the POI and E3 ligase can be quite critical. What would be interesting would be getting a structure of a ternary complex which we know is forming but doesn't form degradation, and a very similar linker where you are getting that degradation. So by comparing these two structures, you can see the slight different nuances of the linker, the direction or orientation of the linker, the angles, for instance, which can give you degradation, or even by optimizing the linker and the angles, you can actually increase degradation. So these are sort of the aspects you need to worry about. Plus also, we know PROTACs, you've got two small molecules of linker. These are big compounds. So you have to take into account, a large compound's solubility, oral availability as well. So all these things play into how, the teams will look at the structure and then feed back through to the chemistry and biology to drive that PROTAC candidate drug. Hope that sort of helps.
Thank you, Paul. We got one more question. I noticed a GPCR construct with an AVI tag. Could you briefly introduce the biotinylation of GPCR? Is there any difference between GPCR and soluble protein?
Thank you so much for the question. So actually, we developed the biotinylation on the AVI tag similarly for the GPCRs. So usually because we never know which orientation of a GPCR is good for GPCRs immobilization onto the chip. So we usually test the AVI tag on both sides of the GPCRs. So according to us, due to the complication of memory proteins like GPCRs, we recommend using a tight binders for immobilization, like the AVI tag. But there are also cases that we can use the CM5 chip by immobilizing the antibody of the GPCR to grab the molecule of GPCRs for detection. So basically there's no fundamental difference between the GPCR and soluble proteins with regarding to the AVI tag biotinylation. Thank you.
Jerry, can you also mention, because sometimes we've also AVI tagged nanodiscs, haven't we? Instead of actually tagging the GPCR itself, for some clients who wanted screening, we tag the nanobodies instead, or so the nanodiscs.
Yeah, that's also another strategy. So we don't really directly tag the GPCR by itself. So if we can reconstitute the GPCR into nanodiscs, we can also consider making the AVI tagged MSP for immobilization. So there is also another option for doing SPRs. Thank you.
Thank you so much. We don't have more questions so far. Thank you for joining our webinar today. If you have any further questions, please feel free to contact with us. You can also check the webinar recording on our YouTube channel and the brand website. Thanks again, and we look forward to seeing you at our next webinar.